Today we will talk about the circulatory system and the blood component and vessels. The diagram shows the anatomy of heart and the anatomy of the vessels and the closed systems belong the mammalian. We can talk about the component of the blood and understand of the structure function such as leukocytes, erythrocytes, thrombocytes, plasma. And we will talk about our understanding about the hemoglobin, oxygen dissociation curve, and I will go to understand the bore shift, compare the differences between arteries and veins. Blood. As we know, the blood in the adult contain 5.5 to 6 liters of the blood, and it's composed of 79% of water. The role of in homeostasis, that's mean, helps maintain pH, temperature, and osmotic pressure. The blood will support growth via distribution of hormone and nutrients. And the involved waste removal such as nitrogen, waste, carbon dioxide. Transport of cellular components around the body to distribute oxygen, support homeostasis, and maintain immunity. If we look at this point, they talk about the importance of the blood to our health and to our body. It is like the conducting, like or communicating methods between other organs, between organs to other organs. Even it is just conducting, and moreover, they transport the materials such as nutrients and hormones. And when the hormones is they secrete in the glands, how can they transport to other bodies or to other organs? By the blood. And the importance of the blood, can be a translocate of the materials such as the oxygen, carbon dioxide, towards everywhere to the body and the organs. Moreover, the contents of the blood, such as the cells, when we see the cells, such as leukocyte, that means white blood cells. It is involved with the immunity and the health. How can the body react with the antigen, for example, or with the foreign materials? By the blood, they transfer all these cells to the target cells or to the problem cells. Okay? What are the components of the blood? If we separate the blood, we can see deposit of the cells and something like watery thing. Cellular components that's composed of erythrocytes, that means RBCs, thrombocytes, platelets, and leukocytes. while non-cellular components is composed of plasma. The plasma mainly water, proteins, glucose, electrolytes, hormones, salts, lipids, and contain, moreover, the cellular components. This picture shows the anatomy of the vessel wall, such as the outer membrane called tunica adventitia, and the middle of this is tunica media, while internal layer is called tunica intima. This the blood vessels anatomy or transection. When we do transection for the blood vessels, we can see these layers of the for the vessels wall. And we can see red blood cell and white blood cells and the platelets. Erythrocytes. It is we can see erythrocytes. How can they? It's moved from the vessels because of features of this erythrocytes has a flexibility to make like a shrink or to minimize their size to move from the vessels, and it stay and it has a hemoglobin. This hemoglobin, look, very important to make transfer the oxygen. Why? Because they react with with the oxygen. The hemoglobin has reaction very. They meant to react with the oxygen so that the hemoglobin can transport the oxygen to everywhere. The nucleus of the blood of the RBCs dissolved after maybe days when it is the red blood cells born. So the shape of the RBCs bioconcave, as I said, a nucleate. They don't have nucleus. Their size 7 to 8 micrometer. They don't have mitochondria and ribosomes. Lifespan is 120 days. It's just 4 months only. After that, after when it's the RBCs break down and recycled where to the liver or spleen. So we can see the ferritin in the spleen. Why? Because they have because of the RBCs destroyed there, and all the ferritin will deposit over there. What the function of the erythrocytes? As we said before, it's delivery of oxygen and circulate around the body. RBC, hemoglobin bind to oxygen, as I said before, in the lungs. And it is vanished the carbon dioxide from respiring cells for excretion by lungs. Some carbon dioxide bind directly to hemoglobin to form carbaminohemoglobin. The rest is converted to the bicarbonate, which dissolves in plasma. Oxygen and carbon dioxide diffuse across the plasma membrane of the RBCs rapidly. Small size and biconcave shape increase surface area for diffusion. As I say, the flexibility to move through the blood vessels. Okay. What is they produced or the erythrocytes production? It's produced in bone marrow, as we know, from stem cells. Uh, the hormone EPO, erythropoietin, controls RBC's production. EPO produced largely by the kidney. EPO produced regulated by blood oxygen, and when it is the oxygen levels low in the blood, that means more EPO will produce, and vice versa. Required for RBC protection. What we need as a supplement or foods or vitamins, we need vitamin B12, iron, and folic acids. When we have iron deficiency, for example, or a B12 deficiency, cause maturation failure resulting in large RBCs. Look, while iron deficiency make the RBCs more smaller. That's why in the test, the laboratory test, we find the person who has iron deficiency, his blood cells or diameter is very small and lots of RBCs. Why? To replace the lack of oxygen, because they need oxygen more, and the bone marrow will produce a lot of RBCs. Some of them, they has like a pain in their bone marrow or on the in the bone, because the production of RBCs, they took all the power from this person and make bone itching or bone act. Okay. What about the blood group or RH group? APO, all the people in the world has either A or B, or they don't have neither A or B. We can call O or zero. Four possible blood groups. A blood group is determined by the presence or of absence glycoprotein or glycolipids on the membrane. Important when considering blood from a transfusion, for example, someone with a type O blood cannot have type AB blood transfer. What do you think will happen if we make a transfusion of the blood with the wrong person? What do you think? That's a question to you.
The platelets and white blood cell will accumulation
Mhm.
on the blood vessel,
Yeah.
and the person will die.
No. Look, they make like antibody antigen reaction,
Yes.
because it is different with the RH. That means that the things foreign enter to the body. All the body, the body will make antibody against all the blood that enter to this person. So that will die because of that. Okay? The accumulation because of that. You said white BC will accumulation. Why is the accumulation? Because they make like antibody antigen. When it is the antibody will come, they surround all these RBCs with the wrong RH. Okay? Blood group. Red blood group type. We see here A, B, and possibility of AB or 0. Antibodies in plasma, look, anti-B, that means belong A. And B has anti-A, while AB don't have either or. While O has anti-A and anti-B, so that we can say it's donor. Antigens in red blood cells, it is A here, while the antigen in B, it is B. A and B antigens here in AB. In O, none. A blood type compatible in an emergency. What will give the person who has A blood group? We can give him A or A, O. Why? Because there is no antigen against to the O, and we can give him O, right? And we can, already he has a antigen, so that's no problem. With a B, we can give them B and O. O, very lucky. AB blood group. He can accept all of them. Universal recipient. While O, poor. Universal is donor, just O. That's a problem. Hemoglobin. Look, this is the hemoglobin. What we can see? We can see the beta chain of hemoglobin and alpha chain. And between of them, they connected with them with the iron, and the iron is surrounding with the heme. 250 million hemoglobin molecules per RBC. Each hemoglobin molecule binds with the 4 oxygen molecules. Each RBC can therefore carry 1 billion oxygen molecules. We can calculate this equation. Hemoglobin contains iron and red pigment, this heme. When an oxygen binds to the hemoglobin shape is slightly altered, making it easier for the next hemoglobin to bind, positive cooperative binding. This means that in oxygen-rich area, like the lung, HB has higher affinity for oxygen, which promotes oxygen binding. In oxygen-starved area, like muscles, you know what is starved area, and that's the area lack of a lot of oxygen. They need more. Okay? فقيرة starved. Starved that mean يعني صايمة المنطقة. Starved. Hemoglobin has a lower affinity for oxygen, which promoted unbinding oxygen. This is good as oxygen offloaded into the muscle. So that's when we do the practice in the gym or workout, as they say, you can consume a lot of oxygen, and a lot of oxygen will go directly to the muscles. Okay, now we can see or study the saturation curve, oxygen saturation curve. The oxygen dissociation curve is S-shaped to positive cooperatively. So here we can see saturation in percentage, while here the oxygen, the amount of oxygen, so they drew the curve in oxyhemoglobin dissociation curve. There is a low saturation of hemoglobin when the oxygen level are low. Hypoxic tissue, so hemoglobin releases the oxygen, sorry. In this area, look, this is the 50 percentage of saturation. Down, it is low or unloading affinity, while up affinity or unloading. So, carbon dioxide and temperature and the other things is that affected to the saturation of oxygen. When oxygen levels are high, i.e. in the lung, hemoglobin saturation is high. So oxygen will bind. This area will oxygen bind to the hemoglobin. Look at this area. When it is the oxygen high. While in the oxygen is low, the oxygen is affinity to unbonding to the hemoglobin. P50 is partial pressure of oxygen at which the hemoglobin is 50% saturated. 25 milli Hg. That's mean the يعني زئبقية. So they make comparison between right shift and the left shift, and this area is wrong. It is normal. When it is area normal with the 50% of saturation, the affinity of oxygen to connect with the hemoglobin is high and is good. Here it's very low, and when it is anemia happen, and it's the temperature that affected with the affinity of the hemoglobin with the oxygen. إذا عندكم سؤال قولوا. Okay. Oxygen saturation curve, the Bohr shift name. Here, left shift, decrease temperature, decrease 2 to 3 dpg, and decrease hydrogen, decrease of carbon oxide. Right shift, reduced affinity, increase temperature, increase 2 to 3 dpg, and increased hydrogen. It is oxyhemoglobin and saturation. It is P oxygen. The Bohr shift is shift of the oxygen saturation to the right. That way, this right. Right shift of the curve facilitated oxygen delivery to the tissue, while in location with the increase of pp carbon dioxide, respiring tissue, oxygen dissociation from the hemoglobin is favored to facilitate oxygen delivery to the tissue. What that mean? That mean when it is this, the carbon dioxide is rise in the tissue, the affinity of facilitated oxygen delivery to this tissue, make like vice versa. Here they ask what factor caused the Bohr shift. When it is the carbon dioxide is low and carbon dioxide is high. Saturation, it is mean to, with the low, is very high. Partial pressure of oxygen. Carbon dioxide or ppCO2 up, temperature is high, carbon dioxide is acidic, and all of these factors occur in a respiring tissue, facilitating the unloading oxygen from the hemoglobin into the tissue. This effect of the ability to consume oxygen with the tissue. Okay, we can study sickle abnormal erythrocyte cell anemia. يعني فقر الدم البنجي. People with the sickle cell, look at this, the shape of the red blood cell will change to the sickle cell and become as a crescent shape. يعني يصير شكلها مثل هلال. The crescent shape RBC are inflexible and fragile and will block blood. Why it is making that problem? Because it is very fragile, and it is when it is broken will like deposit in the vessels and make a problem like the clots in the vessel. Blood flow is blocked, which can result pain in the area. That's a problem with the sickle cells blocking blood flow. Look here. This is the crescent shape of red blood cells, and it is deposit in the branch of the vessels. The hemoglobin in the sickles shape, it's very fragile, and it will be destroyed and make pain in the organ. Now, the leukocytes. That means what's involved with the leukocytes? Leukocytes involved with the immunity. We have a members of the leukocyte in the blood, or the immunity in the blood. We have a monocytes, eosinophil, basophil, lymphocyte, neutrophil. These numbers varies in the blood and the percentage of them in the blood. In normal people, we can see a lymphocyte, for example, 5 to 6 million. In monocytes,
Sorry to interrupt you. دكتورة هما بس يوصل تفضل وياكم observation اوكي بس عن مود البايو يعني عندك انا ما ادري اذا الدكتور حسام معطاك خبر؟
لا تفضل.
هو البحث عن الدكتور حسام بلغني انه مجرد يعني